go.
I'm Bevan Bornsby on Channel Negative 1 with Defective News. I'm gonna hand it over to my pal, Pan Dulce.
We have some breaking news to tell you. There has been a rise in interest for celiac disease. Now, what is celiac disease, you ask? It's a disease that prevents you to digest gluten. Now, we have a guest here to tell more about celiac disease, the legend, Bill Rye, the celiac guy.
Hello everyone, thank you for having me. I'm here to tell you a little more about celiac disease. Celiac disease is an autoimmune disorder in which a person cannot eat gluten, a protein found in rye, wheat, and barley. The precise cause of celiac disease is not known, but they do know that there are certain genes that play a major role for the risk for it. Also, not only do you need to have some combination of the genes, you need to have some sort of environmental factors, such as eating gluten, pregnancy, or viral infection to activate the disease. When you have celiac disease, your immune system is messed up and it senses the gliadin, which is a protein in the gluten, as a toxin and attacks it, which damages the villi and the small intestine.
Protein synthesis starts in the nucleus with a process called transcription, where the information in DNA is transferred to a messenger RNA molecule. A mRNA molecule is the same as one strand of DNA, except thymine is replaced with uracil. RNA polymers catalyze the information of an RNA molecule by reading one side of DNA and stringing together as a complementary strand of RNA nucleotide.
The mRNA molecule is then transported to the ribosomes to start the second half of protein synthesis called translation. At the ribosome, a tRNA molecule reads the mRNA codons, which are sequences of 3 nucleotides which forms genetic code in an mRNA molecule, and creates complementary anticodons to match. As the tRNA molecule is working, it's creating a polypeptide chain that will become a protein.
There are many genetic variations that can play a role in the development of celiac disease, such as HLA-DQ, CTLA4, IL-2, and IL-21, and MYO9B. Even if you have some combination of these variants, you still need an environmental factor to develop the disease. So one example of a genetic mutation that you can have is in the CTLA4 gene, which is located on chromosome 2. Position number 49 is mutated to an A allele. CTLA4 is a protein receptor that functions as an immune checkpoint and downregulates immune responses. So when the base is changed, it causes a difference in the protein, which contributes to the risk of getting celiac disease. In a normal person, T cells fight disease in the body, but in celiac disease, T cells are triggered by gluten to mistakenly attack the enterocytes, which are intestinal absorptive cells. Then helper T cells signal to B cells, which produce antibodies, which is a blood protein produced in response to and counteracting a specific antigen, to gliadin, which is a part of gluten, which may cause additional damage. In conclusion, celiac disease confuses the T cells and B cells into accidentally attacking and damaging the immune system.
Jennifer Esposito is an American actress and author who is known for her roles in Crash, Summer of Sam, and Law & Order. She struggled with undiagnosed celiac disease for many years. Her struggles finally came to an end when she was filming an episode of Samantha Who. One of her teeth suddenly fell out. She knew she needed to get help, but she found herself being treated for mental illness. She says at the time her hair and fingernails were all falling out. A little while later, she got the diagnosis that she was suffering from celiac disease. She changed her diet completely and got rid of all forms of gluten. She says now that food is something that heals her, even though before it was quite literally killing her. Neither of Jennifer's parents had celiac disease. Maybe they had some of the genes, but an outside force never activated it.
All of these genotype examples are based off of the result of an experiment, including 101 French Caucasian people with celiac disease and a control group of 130 French Caucasian people without celiac disease. Also, celiac disease genetics are based on risk factors, meaning if you have, in this case, AA genotype, you have an increased risk of getting the disease. You do not just have the disease if you have a certain genotype. One last thing that has been mentioned many times is that there are many different genes that can possibly contribute to the risk factor. In this case, we are showing CTLA4. For example, if you have an AA genotype, you are 2.36 more likely to get celiac disease than if you have an AG genotype. So if your parents have AA and AG, you have a 50% chance of having a child with AA and a 50% chance of AG, meaning that they have a 50% chance of being 2.36 more likely to develop the disease. For the second example, you have a parent with AG and a parent with AG. You have a 25% chance of being 2.36 more likely to develop celiac disease. You have a 50% chance of getting AG and having the same risk. You also have a 25% chance of getting GG, which means your risk is 1 out of 5 of AG, which is really good.
The only treatment for celiac disease is to follow a gluten-free diet. For most people, this will stop symptoms and heal existing intestinal damage. As long as gluten is not present in the body, nothing will happen.
There are some possible cures that are being tested right now. One is INN-202, which is a drug that uses a potent digestive enzyme to break down the gluten before your immune system can react to it. There's the NextVax-2 vaccine, which aims to immunize individuals against the disease, although it is genetically tailored to those with the DQ2 gene, which is one of the possible genes that increases celiac disease risk. And then there's the BL710, which is a polymer that binds the gluten protein and does not allow it to be broken down.
Thank you, Bill Rye, for informing us about celiac disease. And thank you for watching Defective News, Channel -1.